This is Enzyme and Antibiotic Resistance by Mia and Keely. I'm Keely and I'm going to be talking about our experiment. The experiment question was, how does pH affect the activity of rattlesnake venom as an enzyme to deactivate its target? First, let's start with background information. What are enzymes? Enzymes are proteins that speed up reactions in the body. Now let's talk about the 4 macromolecules. There's carbohydrates, lipids, proteins, and nucleic acid. Let's start with carbohydrates. The monomer of carbohydrates are monosaccharides or simple sugar. The polymer is polysaccharide, and carbohydrates are the body's primary source of energy. Now let's talk about lipids. The monomer of lipids is glycerol and fatty acids. The polymer is phospholipids, and the function is acting as signaling molecules. Now on to proteins. The monomer of proteins is amino acids. The polymer is polypeptide, and it's crucial for most bodily functions. Lastly, nucleic acid. The monomer is a nucleotide. Polymer is DNA and RNA. Nucleic acid is used to store and transfer genetic information. Now let's talk about protein folding. Starting with primary, this contains all info for higher orders of structure. Now let's talk about secondary. This is where large numbers of such local interaction form 8 helices and 3 pleated sheets. Now tertiary. This is a more compact and 3D structure. Lastly, quaternary. This is found in proteins of 2 or more interacting polypeptide chains or subunits. Did you know each amino acid has its own chemical behaviors? Now back to our experiment. Our hypothesis was that the more basic the pH, the faster the enzyme deactivates its target. Our control groups for this experiment was 1 gram carb, 2 gram enzyme cutter, 25 milliliters of water, 3 milliliters of Benedict, and that equaled a pH of 9. Now let's introduce the experiment groups. In bleach, there was 1 gram carb, 2 gram enzyme cutter, 25 milliliters of water, 1 milliliter of bleach, 3 milliliters of Benedict. The starting pH was 11 and the after pH was 8. Now let's talk about the vinegar. There was 1 gram carb, 2 gram enzyme cutter, 25 milliliters of water, 1 milliliter of vinegar, 3 milliliters of Benedict. The before pH was 2 and the after pH was 4. There were 5 constants in the experiment: 1 grams of carb, 2 grams enzyme cutter, 25 milliliters of water, 3 milliliters of Benedict in the boiling hot water. The independent variable in this experiment was the pH, and the dependent variable was the time. To conclude the experiment, we found out that there was 4 minutes for a full color change in bleach and 7 minutes and 40 seconds for the full color change in vinegar. To conclude, our hypothesis was right. The more basic, the faster the enzyme deactivates its target.
Hi, it's Mira Stovsky, and I'll be talking about how antibiotics work in the body. Many antibiotics are made of non-pathogenic bacteria called acetamides, which can be found in soils, deserts, oceans, or plants. Antibiotics can be bacteriostatic or bactericidal. Bacteriostatic means to stop from replication, and bactericidal means to kill the bacteria. The first demonstration we'll be showing is bacteriostatic and how bacteriostatic bacteria interferes with processes needed to multiply, such as DNA replication, metabolism, and protein production. Once the pill has been swallowed, it is then broken down by the stomach acid and absorbed into the bloodstream. Once the pill has made it into the bloodstream, it is able to be transported through the body to reach the site of infection. There are different ways an antibiotic can kill or inhibit a bacterial infection. In this example, a bacteriostatic antibiotic was administered, which blocks the reproduction of bacteria's DNA. This is why our patient feels so much better. Next, we'll talk about how bactericidal antibodies work. Beta-lactam enzymes are bactericidal, which means that they kill the bacterial cell completely. The bactericidal antibiotic inhibits the synthesis of the peptidoglycan layer of the bacterial cell wall. The peptidoglycan layer is important for the cell wall's integrity, especially gram-positive organisms, which I will explain more in depth later. Here we can see how well the bactericidal antibiotic worked due to it breaking down the cell wall, which later caused the bacteria to dissolve. Here I'll explain gram-positive and gram-negative bacteria. Gram-positive and gram-negative bacteria are classified by how they react to a gram stain. Gram-positive has a thicker cell wall with about 30 layers of the peptidoglycan. The cell wall surrounds a single plasma membrane. Gram-negative bacteria has a thin peptidoglycan layer, which is sandwiched between two lipid bilators called diderms. Here's how to tell gram-positive and gram-negative apart. Use a crystal violet liquid, then gram-staining destaining solution, then saffron, and then fuchinase. To get your results, gram-positive will remain purple and gram-negative will turn pink. The reason for this is that the gram stain detects peptidoglycan. The antibiotic we chose to make was synthamicin. Our antibiotic is classified as synthetic lipopeptide because it is a lipid that is attached to a peptide chain. Thanks for watching.